right. Good morning, everyone. Thank you to Lan for the introduction. Thank you to Sid for organizing this mini symposium. And today I will tell you what we are interested in my lab. And the ubiquity in proteasome system has been introduced already by Avram, so I will not go through. Just to remind you that of 20,000 genes in the mammalian genome, about 1,000 are dedicated to the ubiquity proteasome system. Of these, 600 encode ubiquitin ligases. And ubiquitin ligases come in different families. What we study in our lab is the family of Carlin ring ligases. These are multi-subunit ubiquitin ligases, and there are more than 200 members of this family. So what are the CRLs? Carlin, because there is a Carlin subunit in all CRLs. This is a sort of scaffold protein which holds at the C-terminus the ring finger protein. At the N-terminus binds an assembly factor which brings the substrate receptor, which binds specifically to downstream substrates. The ring finger protein is necessary to bind the ubiquitin conjugating enzyme, or E2, which brings the ubiquitin to the substrate. So the family of CRLs is subdivided in 5 subfamilies, each one containing a different Carlin scaffold subunit, and each Carlin will bind different assembly factor and substrate receptors. So for instance, Cal1 will bind a family of about 70 F-box proteins in mammals, and Cal2 will bind VHL box proteins, Cal3 will bind BTB proteins, Cal4 decaf proteins, and finally Cal5 SOX box proteins. And today I will tell you a little bit more about CRL1s that are also known as SCF, as mentioned by Avram, and CRL4s. And again, as mentioned by Avram, UPS controls virtually every cellular function. Its deregulation is involved in many human diseases. And in our lab, we study mostly cancer. So there is a lot of talking about translating basic science, but clearly a problem with translation are often due to bad basic science. So to translate something, it requires a deep understanding of molecular cellular mechanism that has to come before translation. And you have to translate only worthy text. If you don't have a worthy text, translation becomes useless. So this is the importance of basic science, the importance of understanding what the enzymes, in our case, ubiquitin ligases, do in the cell before we can translate anything. And so in our lab, we are interested in molecular mechanisms, and their study allows us to answer 3 major questions. So first of all, what are the CRLs whose aberrant activity contributes to human diseases, such as cancer? How can we exploit this information in therapy? And how can CRLs and other ubiquitin ligases be reprogrammed to target new substrates? So along the years, my laboratory has matched about 40 substrates ubiquitin ligase pairs. And we start in general from proteomics and somatic cell genetic studies to identify the initial hits. And then, of course, we validate them in cell system as well as in animal models. So I'll give you some examples of questions. These are recent questions that we have asked, that postdocs in the lab have asked. KIP1 is a ubiquitin ligase. It's actually a substrate receptor of a CRL3 and is lost in lung cancers. And when it's lost, promotes metastasis. But how? Nobody knew about that. Moreover, when KIP1 is lost, it promotes a Brackness phenotype. So it mimics a sort of DNA damage repair defect, and by which mechanism was not known. Another postdoc asked which CRLs are downregulated by DNA methylation, suggesting that they might be tumor suppressors. Which CRLs provide resistance to PARP inhibitors? And finally, this is a very basic question, which CRL targets D-type cyclins for degradation? So these are questions that different postdocs asked in the lab in the last few years. And this cartoon shows the answer to these questions. Each of these little cartoons is several years of work of a poor postdoc in my lab. So, but we don't stop here. After we understand the molecular mechanism by which some proteins are degraded, then this will give us insights in terms of therapy. And so we'll offer the possibility to understand whether we can exploit this information in terms of therapy, and particularly in terms of how this mechanism can induce sensitivity or resistance to different therapeutics. And of course, we test these once again in cell cultures as well as in mouse models. I would like to go a little bit deeper for one of these particular studies, and I will go through how we discovered that DCAF3, also known as AMBRA1, targets D-type cyclins for degradation. So, as you heard from Abram, we are interested in cell cycle control, and I'm sure that everyone here knows that different cyclins will activate different CDKs at different stage of the cell cycle to promote progression through the cell cycle. And the first cyclin to be activated in the cell cycle is cyclin D1. The synthesis of cyclin D1 is stimulated by mitogens, and once cyclin D1 is so synthesized, will bind CDK4 as well as CDK6, particularly CDK6 is mostly expressed in hematopoietic cells, and will form an active kinase which will trigger cell cycle. Now, there are inhibitors of this kinase, and they're used in the clinic, particularly in breast cancer. So, because of the importance of cyclin D1, many labs along the years have asked whether, what is the system, how cyclin D1 as well as its paralog cyclin D2 and D3 are degraded during the cell cycle, by which mechanism, by which ubiquitin ligases. And these are papers that have been published between 1998 and 2012. And as you can see, they've been published all in good journals, including Nature, Cell, Cancer Cells. And basically what these papers suggest is that cyclin D1 is degraded via one of several CRL1s. Okay? So in fact, if you look at the literature, there are as many as 8 different FPOX proteins have been reported to target mostly cyclin D1, but also cyclin D2 and D3. So there are 8 different FPOX proteins, there are 8 different CRL1 complexes. The problem came when Keiichi Nakayama, a good friend and colleague of mine in Japan, published this paper. And so what Keiichi had shown in this paper that either knockout or knockdown of single F-box proteins or F-box protein in combination of 2 or 3 have really no effect on cyclin D1 degradation. So to be honest, we knew about that because we could reproduce these results. But before this paper was published, I could not convince anyone in my lab to work on ubiquitin ligase for cyclin D because they told me that this was already published. But after Keiichi Nakayama published this paper, I was able to convince Daniele Simoneschi to work on that. And so we started from the beginning. So one of the very first experiments, by the way, each of these experiments has been done in multiple cell lines, but for simplicity, I will show you only representative results. So in these cells, we express dominant negative CAL1. We can induce it with doxycycline. And when we induce CAL1, we stabilize these two known substrates of CRL1 ligases. You heard about P27. We also published with Avram that P21, a cousin of P27, is also a substrate. But despite the accumulation of these established substrates, we saw no effect on D-type cyclins. And this is true also when we enhanced the degradation of cyclin D1 with DNA damage. Again, no effect when we induce dominant negative CAL1. So in a way, this simple experiment suggesting CAL1 doesn't play any role, SCF, CRL1s don't play any role, increasing doubts about all these studies that I have shown you before. But is cyclin D1 a target of the proteasome? Yes. You see, in different cell lines, you treat cells with MG132, blocking the proteasomal activity, and you accumulate cyclin D1. You can also use this other inhibitor. This is an inhibitor of nedylation. Nedylation is a post-translational modification necessary for all CRLs to be active. When you block CRLs, you also accumulate cyclin D1, okay? And you stabilize, this is the half-life of cyclin D1. So we confirmed that it must be one of the 230 CRLs, but before we understand which one, we asked if it's not CAL1, which CALin is involved. So we downregulated by sRNA all CALins individually. We saw no effect with CAL1, no effect with any other CALin. Only when we downregulate CAL4, then we see accumulation of cyclin D1, as well as the phosphorylated form of cyclin D1. It is known that cyclin D1 is phosphorylated on threonine 286. When it's phosphorylated, it's unstable. This is a typical, this amino acid is typically mutated in cancer to stabilize cyclin D1 and to have more cyclin D1 to promote cell growth. So we thought, okay, if it's not CAL1, it must be a CAL4-dependent ligase. Now, CAL4 has another common subunit, DDB1. This is a sort of assembly factor. If you downregulate DDB1, you also stabilize cyclin D1, cyclin D3, and you will accumulate the phosphorylated form of cyclin D1. The conclusion of this experiment is we don't believe that CRL1 has anything to do. It must be CRL4. So we need to understand which decaf among the various decaf proteins present in mammalian cells is doing the job. And so here we decided to use 3 approaches. One approach was very simple. We downregulate by sRNA all the decaf proteins in the cell. But we also wanted to go further and see whether we got same results with 2 other completely unbiased approaches. So let me tell you very quickly. about the three approaches. The first one, we expressed fluorescent cyclin D1 at low levels, and then we treated cells with an sRNA library, sRNA that targets all the decaf proteins into different cell lines. We see accumulation with few of them. One that was very robust was AMBRA1, decaf3, where we saw accumulation that stayed with time, in contrast to, let's say, ERK8, that first accumulated, then tend to go down again. Then we purified cyclin D1, either wild type or the mutant that is not supposed to bind the ubiquitin ligase, and we did it in presence of MG132 or MLN4924, which we know that by blocking the ubiquitination or degradation, stabilize the complex between the ubiquitin ligase and the substrate. And so when we IP'd cyclin D1 wild type, we saw a number of ubiquitin ligases, but the one that behaved the best, as expected in a way, was AMBRA1. AMBRA1 only comes down with the wild type, not with the mutant, and the binding increases in presence of proteasome and CRL inhibitors. Third approach was another unbiased approach in which when we express cyclin D1 fluorescent, then we treated these cells with a CRISPR library, and we saw two candidates. So from these three approaches, there was a common one, AMBRA1, which seemed also to behave in a way that suggested that AMBRA1 was the right one. And we validated in every possible way. This is a simple experiment of sRNA. We downregulated a bunch of ubiquitin ligases that have been proposed or they came out from our screens. Only AMBRA will induce accumulation of cyclin D1. Busy slide just to tell you that in different cell lines we can knock out cyclin D1, AMBRA1, and compared to parental cells, we see a huge accumulation of cyclin D1, D3, and phosphorylated form of cyclin D1. And then we used an acute downregulation of AMBRA. I think this is really the golden standard to understand if a substrate is degraded by a particular ubiquitin ligase. And what we did was to fuse the endogenous AMBRA with a minimal auxin degron. Auxin is a plant hormone that induces degradation of downstream targets, working as a molecular glue that was discovered by Nick Zhang. And this system is used to downregulate proteins in mammalian cells by treating the cells with auxin. So once you treat mammalian cells that have been so modified, you see degradation of endogenous AMBRA within 30 minutes. And once AMBRA is downregulated, cyclin D1 immediately starts to accumulate. You can wash out auxin, and now AMBRA will accumulate again, and cyclin D1 levels will decrease again. And this is just to show you that the effect of auxin is very similar to the effect of MG-MLN. So there is no additional increase when you treat cells with auxin plus proteasome inhibitors or CRLs inhibitors, suggesting that AMBRA is the only, or at least the very major ubiquitin ligase for D-type cyclins. Without AMBRA, you lose the ubiquitination of cyclin D1. And of course, by adding a flag in addition to the MID on the endogenous locus, allowed us to purify AMBRA, and together we co-purify cyclin D1. So this is all endogenous. So the conclusion of these experiments is that CRL4 AMBRA1 target cyclin D1 for degradation. This was done in cells. We did it also in animals. These animals die in uterus, but they die with high levels of cyclin D1 compared to wild-type animals. Then what about cancer? Cyclin D1 is an oncoprotein, so AMBRA1 is supposed to be a tumor suppressor. In fact, we see many mutations and deletions, and a lot of downregulation by promoter hypermethylation. So when you lose AMBRA, you will accumulate cyclin D1 and promote oncogenic events. I don't have time to show you all the number of experiments that we have done, some of which in collaboration with Luca Busino, that is here in the audience, an ex-postdoc in the lab who is now an associate professor at UPenn. I just would like to show you this single experiment. So this is a mouse model of lymphomogenesis, and animals are treated with CDK4/6 inhibitors. And so we use two different doses, low and high, and there is not much difference among the two. In both cases, the animals survive longer. This is using wild-type cells. However, if we inject in mice AMBRA knockout cells, now we see a completely different story. We see that the low dose here has no effect, really like vehicle. And then the high dose, yes, will increase survival, but not as well as when you use wild-type, AMBRA wild-type cells. So the absence of AMBRA reduces sensitivity to CDK4 and 6 inhibitors. So in conclusion, we found that CRL4 AMBRA controls cell cycle progression by inducing the degradation of cyclin D1, and deregulation of this event will induce embryonic lethality, oncogenesis, and resistance to CDK4/6 inhibitors. So in the course of this work, we had an observation. We saw that cyclin D1 is present in the nucleus of some cells, and these cells happen to be EDU negative. So either cells incorporate EDU, either they replicated their DNA, or they express cyclin D1, but never both. So cyclin D1 is eliminated prior to DNA synthesis. In parental cells, if you remove AMBRA, now will be present in all cells. Okay? And the question is, why mammalian cells need to eliminate cyclin D1 before S phase? And this is an important question because cyclin D1 has been known for 30 years to bind pCNA, an important cofactor in DNA replication as well as in DNA repair. And to make a long story short, I will only show this cartoon. These are unpublished results. But basically, what we know is that in S phase, you require mismatch repair. This is because DNA replication has makes many mistakes, and so mismatch repairs will repair them. If you have cyclin D1 during S phase, as we saw in AMBRA knockout cells, but also in many cancer cells, cyclin D1, through a mechanism that, if you are interested, I will tell you later, is inhibiting mismatch repair. Therefore, you will have deficient DNA repair and genome instability. This effect is totally CDK4 independent. So cyclin D1 doesn't need to bind CDK4 to block the activity of mismatch repair factors. In fact, it needs to bind P21. And this suggests that perhaps cyclin D1 degraders could be more effective than CDK4/6 inhibitors, because with these guys, you only block the activity, and therefore the effect of cyclin D1 on cell cycle progression, while with a degrader, you will remove the activity, but you will also remove the effect of cyclin D1 on genome instability. So in the last few minutes, let me talk you to introduce actually products and molecular glues. About 70% of disease targets are undruggable via classical approaches, and so people are thinking about targeted protein degradation, and in particular, they want to use products and molecular glues. As you can see from the font size, I'm biased towards molecular glues. But let me tell you what is a product, although I'm sure that almost everyone in this room knows. Products are proteolysis-targeted chimeras, and they are supposed to target undruggable proteins. And what they do, they are bringing together an ubiquitin ligase with a substrate that normally in the cell doesn't bind that particular ubiquitin ligase. And it does it through a product which is formed by two warheads, one binding the ubiquitin ligase, one the substrate with a linker in between. And so products work with a non-stoichiometric catalytic mechanism of action, which should allow to get rid of the substrate by using doses that are lower than the doses that you will use just by inhibiting, binding and inhibiting the neo-substrate. So you can target theoretically any ligandable proteins. So you have a compound that binds a pocket of the substrate, you can form a product. And in general, we believe that these are very effective on proteins with limited resynthesis rate. We're concerned, and there is some evidence that if there is too much synthesis, you might overwhelm the ubiquitin ligase, and therefore you don't get enough degradation. Now, what about molecular glues? Molecular glues, in contrast to products, involve a non-chimeric small molecule, so this is much smaller than a product, and like products, they will allow new interaction between a novel substrate and UBL. They also have a catalytic non-stoichiometric MOA, but it has some advantages because it's a small chimeric, non-chimeric protein. It's also small enough to be a drug-like compound, and therefore, might not have the disadvantage of the product to be large and therefore limit cell availability, increase cell toxicity. Toxicity in the patient. I apologize. The other interesting part is the studies from Ning's lab, and I believe he will present some of these, shows that you don't need high affinity on both sides. Here you do. Here you need affinity only on one side. And in general, the substrate, but it could be the opposite, the substrate doesn't have a pocket that is ligandable. The binding occurs when the small molecule binds the E3 with high affinity, and then it forms a new surface which is recognized by the downstream substrate. So in contrast to product, which can target untargetable proteins, molecular glues have the advantage also to target unligandable proteins. And finally, most products work with one or two UBLs. In the case of molecular glues, many ubiquitin ligases can be used, allowing a substrate-centric approach rather than a ubiquitin ligase-centric one. Now, the great advantage of products is that allows rational designs, while molecular glues, not many people know how to make them. And in fact, the identification of molecular glues is a very challenging question in the field. And I use this word de novo because there are indeed molecular glues in nature. I mentioned plant hormones that were studied by Ning. These are nothing else than molecular glues bringing substrate to CRL1, like ubiquitin ligases. And there are drugs that have been used for decades in the field, and people didn't know that they were molecular glues, and only more recent studies had shown their mechanism of action. So, imides, sulfonamides are molecular glues that target new substrates to CRL4s. So to summarize, what we study in the lab are the fundamental molecular and cellular mechanisms regulated by CRLs. We use many different approaches to study these enzymes, and we do study these enzymes because we are curious, but also because we think that molecular mechanisms explain disease mechanisms and inform about vulnerabilities and how to target them. And so I stop here, and these are people in the lab, some of the recent alumni in academia, Luca, which you will meet this afternoon, and some of our collaborators. Thank you for your attention.